So I was 3 years old when my mother and I were outside one day watching ants crawl around on the pavement. And she said to me, "Megan, you really like bugs, don't you?" And I said, "Yes." And she said, "Did you know that entomologists are scientists that study insects?" And I said, "No, but I want to be an entomologist." And what fascinated me most about insects then, as well as now, is how such tiny and seemingly insignificant creatures can, through their many numbers, have huge impacts on our world. And most often that impact is positive, but in the cases of mosquitoes and the diseases they transmit, the impact is decidedly negative. The World Health Organization estimates that over 750,000 people die from mosquito-borne illnesses every year. And this includes diseases like malaria, yellow fever, dengue, Zika, and West Nile virus. And the truly tragic part is that the majority of the people that are infected and die from these diseases are children under the age of 5. Now, as a mom of 2 small children myself, I can't even imagine. And of course, you all know that we become infected with these diseases when a female mosquito lands on us and takes a little bit of our blood and leaves behind a parasite that makes us sick. So I was teaching a field camp for inner city high school students about insects. I told you, I really do love bugs. And I told the students that it's only female mosquitoes that bite us, and that they do so that they can make eggs. And one very enthusiastic and insightful young man said, "Hold up, hold up, hold up, hold up, wait. Every time a female bites me, mosquito bites me, I'm getting her pregnant?" And I had to laugh just like you. And I could totally see why he would think that based on the information I had given him. But no, there are male mosquitoes that fertilize the females. And so when the female bites us, she's not after our genetic material. Instead, she uses the protein from our blood in order to produce her eggs, which you can see this female is doing right here. She's laying her eggs on or near standing pools of water. And then a few days after laying the eggs, they hatch out into the aquatic larvae, the baby mosquitoes that grow and develop in the water and then eventually emerge as adults to the mosquitoes we know and loathe, and the cycle continues. Okay. Now, something that all of us who live in Ohio are very familiar with is that there are seasons or times of year when mosquitoes are very abundant and we can't go outside without getting bitten. And then there are other times of year, like late fall and winter, when we enjoy a reprieve from these mosquito bites. And this is because during these times of years, the mosquitoes are in a hibernation-like state where they're devoting all of their resources to survival. And because of these changes in mosquito abundance, we also see changes or cycles in the diseases that they transmit. So as you can clearly see here, the number of cases of West Nile virus peak in the late summer and early fall when the mosquitoes are most abundant. And this trend holds true for other diseases like malaria and dengue and Zika, that are in tropical environments. These diseases peak during and shortly after the rainy season when their mosquito vectors are most abundant. So, given this tight correlation between when the mosquitoes are around and when people are getting sick, it's not surprising that oftentimes the best way that we have of controlling these mosquito-borne illnesses is to kill or control the mosquitoes themselves. And typically, mosquito control looks like this, right? Spraying lots of pesticides around where the larvae are living or in the air to kill the adults. And many of you are aware that these insecticides can oftentimes have very negative consequences to other animals in the environment. And even worse, mosquitoes are becoming more and more resistant to these insecticides. And so these measures are even less effective now. Therefore, we desperately need new and innovative ways of controlling mosquitoes so that we can better protect ourselves and break this disease transmission cycle. And there's a lot of great research being done on new ways of killing mosquitoes, but very little research has focused on the seasonal aspects of mosquito biology, which led first my PhD advisor and now me to ask the question, can we trick mosquitoes into thinking it's the wrong time of year? So in the case of the mosquitoes that we have here in Ohio that transmit West Nile virus, trick them into thinking that it's wintertime when really it's summer so that they don't bite us. Or, perhaps better yet, trick them into thinking that it's summer when winter's on its way so they aren't able to survive our cold Ohio winters and we have fewer mosquitoes the following spring. So it might seem utterly impossible that we would ever be able to trick a mosquito into thinking it's the wrong time of year. But we know that mosquitoes are relying on a single cue to know what time of year it is, and that cue is photoperiod, or day length, where long days signal to the mosquitoes that it's summertime, and they devote all of their resources to reproduction, to biting us and laying eggs. And in contrast, short days signal to the mosquitoes that winter is approaching, and they devote all of their resources to survival. So in this case, the mosquitoes don't bite us, they don't lay eggs, and they pack on the fat the same way that hibernating mammals do. So photoperiod, or day length, is how mosquitoes are telling what time of year it is, whether it's summer, spring, winter, or fall. But we don't know precisely how mosquitoes are measuring day length. But just like us, mosquitoes have circadian clock genes that regulate their daily activities. So when they're active, and when they're resting, and when they're mating, and when they're feeding. So it's possible that these same genes that are telling mosquitoes what time of day it is might also be helping them to measure how long the days are, to distinguish a long summer day from a short winter day, and thereby appropriately coordinate their seasonal responses. So to determine whether or not clock genes are involved in measuring day length and in initiating seasonal responses in mosquitoes was the focus of my PhD research. And one of the ways that I addressed this was using a technique called RNA interference. So many of you might remember from your high school biology classes that RNA is a molecule like DNA, and it can serve as a messenger, taking the genetic information that's inside of the DNA in the nucleus of our cells and allowing it to be translated or made into the proteins that perform the functions in our bodies. And RNA interference simply blocks or interferes with this process. And the way that I and other molecular biologists use this technique is to determine the function of a gene or protein by removing it from the system. So to extend the clock metaphor, I use RNA interference to block or suppress individual clock proteins, like removing cogs in the wheels of a physical clock, and then determine whether or not the mosquitoes were able to appropriately respond to seasonal cues. Okay, so there are two steps to getting an RNA interference experiment to work. The first step is to make double-stranded RNA. Hello. There we go. Okay. Which you can pretty easily do in the lab. And this double-stranded RNA is identical to the gene that you're going after. It just has two strands instead of one. The second and more difficult step is to get the double-stranded RNA inside of the mosquitoes. So to do this, I needed a very fine needle, a microscope, and a whole lot of patience to individually inject hundreds of adult mosquitoes. And fortunately for me, the ladies were strong. They survived this whole ordeal, and what I found was really exciting. I saw that when mosquitoes were reared under short-day, winter-like conditions, but I knocked down their circadian clock genes, these females actually behaved like it was summertime and devoted all of their resources to reproduction. In contrast, when mosquitoes were reared under long-day, summer-like conditions, but I suppressed a different gene that was associated with their circadian clocks, these females acted like it was wintertime and devoted all of their resources to survival. So, taken together, these results provide compelling evidence that we really might be able to trick mosquitoes into thinking it's the wrong time of year simply by messing with their circadian clocks. However, there are two major challenges before we are able to implement this work into a viable strategy for controlling mosquitoes. And the first difficulty, which might be less obvious, is one of specificity. So the circadian clocks in mosquitoes are highly similar to the clocks in other insects, including honeybees and praying mantids, which are good insects that we like to have around. So it wouldn't be possible to use RNA interference to mess with or interfere with the clocks in mosquitoes without also interfering with the clocks in these insects, which would have negative consequences for their biology. The second and more obvious difficulty is one of delivery, because clearly you can't go around injecting individual mosquitoes as a way of controlling them. So members of my lab and I are now working on both of these issues. So to address the issue of specificity, we're now using a variety of molecular assays to identify the specific genes and proteins that the circadian clock is interacting with to regulate long day responses that are involved in blood feeding and laying eggs, as well as short day responses that allow the mosquitoes to survive the winter. So we're hoping to be able to open up this black box and identify the specific genes involved in both of these processes. Then, once we've identified these genes, it might be possible that we can again use RNA interference to control mosquitoes. But this time, rather than individually injecting adult mosquitoes, we could put the double-stranded RNA into the water where the baby mosquitoes, the larvae, are living. And in fact, my collaborators and I have done these experiments and have shown that if you put high concentrations of double-stranded RNA with the mosquito larvae, you can prevent them from making the targeted protein. And if we identify the right gene, we might also be able to convince mosquitoes in summertime that it's winter and prevent them from taking our blood and from laying eggs. In contrast, if we're able to identify a different gene that's involved in that's critical to allow the mosquitoes to survive the winter, we might be able to make genetically modified mosquitoes that have a mutation in this gene. Then we could connect this to what's called a genetic drive mechanism to ensure that every time the mosquitoes mate, this is passed on to their offspring. So in this way, we could release a few of these genetically modified mosquitoes in the spring, and the mutation would amplify throughout the natural population so that by the time fall rolled around, none of the mosquitoes would be able to survive the winter. They would all die, which would be amazing. So in conclusion, I realized my 3-year-old ambition. I have a PhD in entomology, and there you can see me on the day of commencement holding 2 labors of love, my dissertation and the first of my 2 children. Although as a new assistant professor in the entomology department, I have a lot of work to do, I hope that today I've convinced you that seasonality matters to mosquitoes the same way that it matters to us. And although it would be utterly impossible to trick one of us into thinking it's the wrong time of year, mosquitoes seem to be more easily fooled. And I hope that the efforts of my students and the others in my lab will one day lead to more effective ways of killing mosquitoes, so that when children all over the world go out and look at bugs, look at ants crawling around on the ground, just like I did when I was little, and just like my daughter is doing there, that they won't have to worry about getting bitten by a mosquito and contracting a deadly disease. Thank you.